since just the day before full moon, and we've been going out each day, about 3 dives per day, starting to do surveys. So beginning day before full moon, and what we do when we do those surveys is we're in the water. We're trying to estimate the number of fish that are there using a couple of different techniques. First and foremost is diver estimates, so people going down and estimating how many individuals they see. But we also have a video pan where we take a high definition underwater video, and we have somebody on a scooter swim the length of the aggregation where the fish are, and they tend to be up in a band, so that when we run the video with the scooter along that band, we can subsequently take that video and we can count how many fish are in it. And that's another way we get at how many fish are there. And then finally, we're doing some tagging techniques. So those are the kind of the key things that we're starting to do, and we can talk a little bit about some of the emerging methods we're using, if you'd like. But go ahead.
Well, so I want to show them a couple of videos from last night.
Yeah, and so
okay?
That's great.
Do you want to set it up a little bit?
Sure. Yeah, last night was the first night that we saw spawning this year, which is always a real treat. The nights leading up to spawning, we see the fish get more and more frenzied, and we think it's going to happen, and we all take bets as to whether or not this is going to be the night. And
are there specific behaviors that you guys are looking for down there that indicate you're like, oh, I think this is going to be the night? Right?
Yeah, there's a couple. One is they on nights leading up to when the big spawn happens, the fish start to get more and more frenzied. So they're moving faster. They're checking each other out. They're trying to figure out where the action is going to be. So they start moving a lot. But the other key indicator is that the fish will what's called hydrate their eggs. So the females are the ones that have the eggs, and they will develop those eggs long before they come to the spawning site. But you can think of it as having a plastic bag that you bought from a grocery store that has deflated balloons in it. Yeah.
Sure.
And then leading up to the night of spawning, the night before spawning, the day before spawning, they will hydrate those eggs. And they use water to blow the eggs up so that they're ready for release. Well, that causes the bellies of those females to get huge, big, distended, fat bellies.
So you see them kind of, I mean, they look funny, and then it's stretch marks and everything.
They get stretch marks. It looks like it hurts. They're so big. It doesn't look normal for a fish. And when that happens, you know that spawning is about to happen. And sure enough, last night, we got down on. They were even spawning before we got to the fish. So we came up.
Paint us a picture. You went out there. What time did you go out?
We go out. We try and the fish tend to spawn starting about 15 to 20 minutes before sunset, which is right around 6:20. So we try and get out there and get in the water around 5:45, and we did that last night. Swam out to where the fish were after dropping off the boat, and by the time we got to where the fish were, you could see great plumes of gametes of eggs coming off the wall as in the distance as we came up.
So you already saw it as you were going in. It had already started.
Yeah, it's amazing. The visibility can be 100+ feet, really clear visibility, and there's so much spawning that happens. It looks like a wall of just clear, like milky water coming at you. So it's so much gametes in the water. It's really impressive. And as we get there, those fish are coming up in bursts. So you'll see them, one female typically, we think, and then many males following her, and she releases her eggs, and they're releasing gametes at the same time. So you see these, it almost looks like fighter jets as they're releasing these trails of gametes and spawn. And so it's a beautiful sight, and hopefully we'll be able to show you guys that.
Well, one thing that comes up that I've had a few students ask about are there predators that show up out there during that spawning? Do you guys see stuff?
Oh, for sure.
Yeah?
So a couple of different kinds of predators. One we worry a little bit more about as divers than the other. We see sharks out there regularly. Now, of course, we've never had a problem with the sharks, but it's always in the back of your mind, right?
A little unnerving.
And so we just last night, I think I saw as many as 4 different sharks, and they're cruising up and down, and they're trying to eat the grouper themselves. And so when those fish are coming up in that big spawning burst, and they're thinking about reproduction, they're not really thinking about sharks, and so sharks will take an opportunity to take a bite.
So it's pretty easy for the sharks at that point because they're so distracted by the aggregation and the spawning that it's kind of easy pickings for the sharks.
It's a perfect opportunity for the sharks. And so the following day, we'll often see many different grouper lying on the ground that have big shark bites out of them.
Really?
Yeah. So
Oh.
They take a big divot out of the population. They're pretty good at catching grouper. That's one predator, and then the other is on the other side of the size spectrum. Those are typically small silvery fishes like mackerel scad. And mackerel scad will come in large schools, and they'll feed on the eggs. So after those eggs get released by the females, here will come a school of mackerel scad, and they'll be picking the eggs out of the water and eating that reproduction as it's happening.
Wow.
And so they're getting it from both ends. The sharks are attacking the adults, and then once the adults release the eggs, these silvery fishes, mackerel scad, will be feeding on the eggs itself.
So between the predators that are there, and then the fact that after the spawning happens, those larvae now have to survive out in the water for like 40 days or something like that before they come back. It seems like there's lots of opportunities for them to get eaten. For sure,
yeah, 35 to 45 days typically is what the larval duration would be, and a tiny fraction of those eggs turn into an actual fish that lands on a reef. Less than 0.001%. It's minuscule.
So I know a lot of you guys have asked, well, how many eggs do they release? And you've said, some of the larger females can release millions of eggs, which seems like a ton. And it seems like we should have so many new groupers showing up every year. But the fact is that the vast majority of them are being eaten.
Yeah. So if you think about it this way, if a female produces 3 million eggs, and if just 2 of those survive to an adult, then you get a population that replaces itself. So you get a population that's not growing, but it's not declining. If you get just three fish out of 3 million eggs that turn into adults, that population is growing by 50% a year.
Wow.
So it is of the millions and millions of eggs that get produced, we're doing well if 3 of those eggs turn into an adult. So it's a huge amount of mortality.
Yeah.
What's cool about this though is, we're talking about sharks and we're talking about silvery fishes, and each of those things being, different parts of the food chain, all feeding on these grouper. And it really speaks to the fact that this aggregation is not just important for Nassau grouper.
Right.
It's important for the ecosystem of the reefs here in the Cayman Islands. It's not just about reproduction. It's about everything else that reproduction supports.
Wow. Okay, so I'm going to share with you guys a video from last night that shows some of those predators going after the Nassau. So hold on one second while I switch over to another screen. Actually, there we go.
So that's a large black grouper there. That's another species of grouper that spawns out there, although we don't typically see them spawning. Now these are Nassau grouper that are spawning, and that's what it looks like out there. Last night, you'll see these large schools of fish. And there you saw a shark. See, that's what they do. Those sharks, that's a Caribbean reef shark, and they'll try and take advantage of that spawning event for the grouper to come and hunt the grouper as they're releasing their eggs. That's cool. Yeah.
So fast.
That's great. I didn't know we got that on camera. And there, see those silvery fish there? Those are mackerel scad, and that's a school of mackerel scad coming into feeding on the eggs after they've been released by the Nassau grouper. And so, that silvery going through there is a barjack, and we have a rainbow runner. So lots of different species are egg predators out there, and rely on those eggs for sustaining themselves. And that's a blackjack, which is hunting on the mackerel scad. So it really supports an entire food chain, that reproduction that Nassau grouper are doing.
Okay, here we're back. Were you guys able to hear Dr. Simmons talking during that video, or were you just able to hear the video? Yeah, you could hear him. So we just saw their predators. The next video I have is of one of the other scientists, Allie. I don't know her full.
Dr. Allie Condelmo.
Yes, and she's got a big net. So we're going to show you the video, and then you're going to talk a little bit about what she's doing.
Sure.
Okay.
That's
for we're going to share.
Okay, yeah. So that's a plankton net, and we, in the marine sciences we use plankton net to do lots of different things, but primarily, not surprisingly, we use it to collect plankton. Now, once these eggs are released into the water, they are technically plankton. They're going to be largely controlled in terms of their fate by where the ocean currents take them. What Allie is doing here is she's using that plankton net in order to collect eggs from lots and lots of different spawning events from the Nassau grouper. And we use those eggs to look at a couple of different things. One is the genetics of the population, which we hope to be able to use to reconstruct how many fish are there, and look at things like parentage, how many males are contributing to the fertilization of female eggs. And then, importantly, we're using the eggs that we collect and taking it back into the lab and raising those up and looking at the effect of things like changing ocean temperatures on the quality of those eggs. So do the eggs survive equally well in warmer temperatures versus less warm temperatures? And how does that warming temperature in the ocean change development of the larvae? And this is, of course, important because we know that in the next 50 to 100 years, in the Caribbean basin, the water is going to warm anywhere from 1 to 4 degrees Celsius. So it's going to warm quite a bit, and there are some pretty stark implications. There's some impacts that warming temperature is going to have on the development of eggs and larvae.
So how does a warming ocean affect? Do we know what do we think the effects are going to be of a warming temperature on those eggs, on those gametes?
Yeah, so first of all, it's important to know that when these Nassau grouper spawn here in the Cayman Islands, they are picking the time of year to spawn that is the coldest water temperature available here at any point in time.
So temperature is very important.
Very important, and they're already spawning at that coldest point in time. So any increase in temperature, average increase in temperature, means that they are not spawning at their optimal temperature window. So now the question is, what does that increasing temperature do? And our preliminary findings suggest that increasing water temperatures both increase probability of hatch. In other words, eggs just straight out die. And when eggs do hatch to larvae, the larvae themselves aren't able to survive as long without feeding. That's called survival to starvation. So that water temperature appears to have some pretty major impacts in terms of egg hatch rate and survival.
So, okay. Do you ever get worried about collecting eggs? Like, we want all those eggs to be out there, don't we? Is that a concern? And I'm only asking that because students always ask me this.
I get that question a lot as well. Yeah. So if we maybe collect a total of 100,000 eggs, I would guess in any given year. That's probably an overestimate. If we think about each individual female releasing 3 million eggs, and there are probably 3,000 females, the amount of eggs that we're collecting is such a tiny fraction that you probably would need to use more zeros than would fit across your computer screen before you got to the fraction of eggs we're collecting. It's a tiny amount. It simply doesn't matter.
And so I know, and I'm going to post some of this on the blog tonight, that at the place that you guys, the researchers, are staying, you've now created a lab in one of the bedrooms, and you took some of the eggs in there last night. What is it that you guys are doing with those eggs?
Yeah, so we are again measuring those impacts in terms of temperature and estimating rates of hatching and survival. What we're doing now this year is we're trying to pair the size of the females, which we're estimating using a thing called stereo video, and I think
we're going to talk about that next.
Great, and then what we can do with that is we can look at whether or not the characteristics of the eggs in terms of their health, their ability to survive and grow, is a function of the size of the females that are releasing those eggs. What we know from last year is that there is quite a bit of variability from one female to the next in terms of the fitness or the health of those eggs. And that's the silver lining in the research so far, because what it suggests is that, yes, by and large, warming temperatures are going to have a negative impact on egg development and survival. But because there's variability in the population, there may well be adaptive capacity for that population to respond. In other
What
words,
is that? Yeah.
in other words, because there are some females that produce eggs that are better able to cope with warmer temperatures, one presumes that those females are going to be more fit. In other words, they're going to be producing more baby grouper that are going to recruit to the population. They're going to show back up as adults. And because those more fit females that produce more eggs that are more robust to temperature are producing the new adults in the population, those new adults will also produce healthy eggs that are more capable of dealing with temperature. That's the presumption. And what that means is the population itself will respond in an evolutionary sense to changing temperatures.
Right.
And in that way respond to a changing ocean.
Okay. Well, before I let you go, could you tell me what's going to happen next?
Yeah, so we're going to keep collecting eggs and keep monitoring the spawning. That's still happening. What we haven't talked about at all is we've got a lot of work going on over in Cayman Brac. So we don't just work on Little Cayman. We have right now, as we speak, we've got a boat and a team of divers doing stereo video, tag counts. We're using video to do things like capture images of individuals and using facial recognition to keep track of individuals through time. So we're doing all of that over on Brac as well. And so that's a big part of our field work. It's a huge logistical challenge, but it's a really important one because what we can show and what we have shown is that the success of conservation and fisheries here in the Cayman Islands doesn't just apply to Little Cayman. It is, in fact, something that's a success across the islands.
Wow. And so what is your sense? You've been coming out here and diving on this aggregation for 20 years. How do you feel when you're out there now and you see all of these fish? I mean, I know when you first went in there was maybe 1,500 fish. And now, I mean, I don't know, that 7,000, maybe more fish out there?
We'll see, right? Yeah. So it's always fun to collect all these data and process it and then get the estimate. But based on last year, we might expect something like 7,000 fish. I tell you what, every year it's always new. It's always fresh. There is nothing as amazing to me as diving on that aggregation and seeing those fish reproducing. It's a sight that's it never gets old. It's just impressive. You see that many large fish that in the rest of the Caribbean today are very rare. Here on this island are in abundance and doing things that they used to do 100 years ago everywhere, and that's really cool. It's a hope spot. Yeah. And if we can replicate what happened here to the rest of the Caribbean, we can rebuild a population of fishes that used to be everywhere in an abundance, and I think we've demonstrated here that we can do that.
Wow, what an amazing thing for the Cayman Islands too. Wow, awesome. Well, thank you so much for being here. I'm going to bring in another scientist who's going to talk to us about the stereo video that we've been shooting that you were just talking about. Yeah.
Yeah.
All right.
Thanks, everybody.
So I'm going to invite Dr. Scott Happel to come over and talk to you guys. He's going to share a camera, and then I've got a little video of what. Oh, Spot Bay, do you guys have a question? Do I see there's? Let's get you up on the screen. There you are. I see you. If you guys, hi, welcome. I'm so glad to see all of you guys here. If you have questions while we're talking, let your teachers know and they can type them into the private chat and I can go ahead and go to those questions. But also know that we'll have some time at the end to do questions as well. So, Dr. Happel, could you introduce yourself to everyone out there and tell us a little bit about what you do on the project?
I sure will, Todd. Hi, everybody. My name is Scott Happel. As Todd just said, I am a professor of fisheries at Oregon State University, and I have been working down here for 15-ish years. Wow. Seems like a really long time. And I've done a variety of different parts of the project. It's all a team effort, and so everybody ends up doing a little bit of everything.
Right.
I've been doing a bunch of the length-based analysis originally. I did a bunch of that.
So size.
Looking at the size distribution of the fish, which we'll talk about when we talk about our stereo camera here in just a second. And I've been helping out a little bit with the larval work, not too much on that, but a little bit. And we're doing a new project this year. Maybe we'll get a chance to talk to about in a minute.
Yeah, and
about some environmental DNA.
Oh, definitely.
If we want to talk
absolutely.
as well.
So about size, why is this important for the project, for us to be studying size?
Yeah, so every year we go out and we try to measure the size, what we call the size distribution of fish, right? How many fish of what sizes? And we use that information to look at how the population is changing over time. So if you can imagine, if over a really long time we only ever saw big fish, well, maybe that's a good thing, but that would indicate that no new fish, no new small fish are coming into the adult population to spawn.
So you actually want to see some smaller fish.
What we want to see is a really broad range of sizes. Okay, but it's really hard to measure fish underwater, of course, because it's hard to swim around with a ruler.
They're moving,
holding it up to the side of a fish like this.
Right, that doesn't work.
It doesn't work so well. And so for many years, we had a system that was basically what we called our laser video system. It sounds kind of cool, right? Who doesn't want to use lasers for science?
Yeah, it's pretty awesome.
Right. And what it was we had a camera, and then on either side of that camera was a pair of lasers that were set exactly 25 centimeters apart. So I could take the video and I would find a fish and I would put those lasers on the side of the fish while it was sideways to me. So the lasers would hit the fish like this.
Right.
And I know they're 25 centimeters apart.
Because they're parallel.
Exactly.
Yeah.
Right. So if I know that much of the fish is 25 centimeters, then I can use computer software to say, okay, well, if that's 25 centimeters, then the entire fish is 54 centimeters or something like that. Okay. And it works really well, but it's a little time consuming because you have to put the lasers on this fish and then on this fish.
Right. You got to do one at a time, individually, and you have to get a photograph of each one of those.
And so this year we're working with some new technology called stereo video. Right.
So why don't, before you talk more about it, I'm going to share a video of it happening.
Okay, great.
Is that okay?
All right.
Perfect. All right. So hold on, you guys, and I'm going to pull up a video of the stereo camera in action.
All right. So this is Dr. Ali Candelmo. And she has the stereo video in her hand. And you can see there's two boxes on the end of it. And I have it here, and I'll show it to you in just a second. Each has a GoPro camera in it. And those cameras are set, in this case, they're 80 centimeters apart, basically 2.5 feet apart. And they're bent in, we call it toed in, like your toes pour in. Toed in at a 5-degree angle. And so maybe the simplest way to explain this, and Todd, you and I were talking about throwing a ball last night, right?
Yeah.
Okay. So it turns out our brains are really, really good at doing complex math. And the complex math I'm talking about is that we have two eyes, right? So we have binocular or stereo vision, right? And our eyes are set a certain distance apart. And so if I'm looking at an object out this far away, my left eye is coming in at an angle a little bit like that.
Right.
And my right eye is coming in at a little bit of an angle like that. So my eyes are toed in just a little bit, right? Let me move my hands in the same, toed in just a little bit to converge on that
one point,
right? And what allows me, say you and I were out throwing a ball, my eyes converge. My eyes can tell how far away you are. And with some complex math based on how strong I think my arm is and how heavy the ball is, I can throw that right to you because I know where you are.
Right.
Pretty cool.
So it gives you almost like a 3D representation. It
gives me a 3D representation in space.
Yeah. And
I could probably tell about how tall you are too.
Right.
So that's hard to do visually underwater. And so this year, instead of doing our laser video and wanting to be able to use stereo vision, maybe I can scoot back here. Let's see if you can scoot that back. This is our stereo camera. We can tilt it this way a little bit.
There we go.
There we go. This is our stereo camera. And it's basically got a box on each end, one there, and these two handles. And we go around like that. And like I said, the boxes on the end are canted in. And so now, instead of having to use that laser, which works okay, and put it on one fish, and then the next fish, and then the next fish, these cameras are towed in so that wherever an object is in space, I can see it in 3 dimensions and measure it directly.
Wow.
So if there's 20 fish on the video while I'm filming it, I can measure all 20 fish at once. That means I've just increased my data collection rate
A lot.
by 20,
Wow.
20-fold, right?
Wow.
And so, and it's more precise. And so we'll generate much better data. And as Dr. Semmons was just talking about, we can use that for our spawning work to see how different females produce different quality eggs. But then we can also use it to track the changes in the size classes of the fish over time.
Wow. That's awesome.
Right. And in fact, we've probably already talked about this with the students, but our initial indication that the population was recovering came not from counting animals, but from the changes in the size distribution. And that was back in 2012. And we're like, wow, we think we're really on the right track here.
And that was just based on the variety of sizes
Based on the lasers that we were doing, right?
Wow.
And then move forward several years when we got better at doing the counting and had a longer data stream, and all of a sudden, wow, it really is happening. But our first indication that recovery was happening was because the size distribution, so does those small fish, were starting to move into the adult population to spawn and create more babies.
That's incredible. We have a couple of quick questions that I think you can help with. And this is from Miss Amanda and Miss Sherilyn's classes at CIS. The first one is, what's the estimated size of the grouper population in Cayman?
This is the best time ever to ask that question because we just published a scientific paper on that Dr. Waterhouse was the lead author on. And the estimate is that the population of adult fish, and I'll clarify that in just a second, the population of adult fish is between 6,000 and 6,500 fish.
Wow.
Okay. So that's how many fish we're seeing just out the aggregation.
And that's just the adults.
That's just the adults. So there's lots of juveniles all around as well that aren't included in that. So many thousands. You
guys actually saw one of those juveniles yesterday when we were on our underwater live stream on Bloody Bay Wall. We saw a beautiful young Nassau right under the boat. So that would be one of those.
And hopefully in 2 or 3 or 4 years, that fish then joins us out at the spawning aggregation site at the west end of the island.
That's so cool.
Yeah.
All right, we have another question, and here's from John Gray, High School. Welcome, John Gray. Thank you for being here. What can be done to increase the population of the grouper?
Well, obviously having what we would consider to be appropriate management in place is the best way to do that. And I would say that because of the willingness of the fishermen to allow it to happen, and in some cases help out, and the efforts in data collection through REEF and the Department of Environment, and the changes in the conservation laws that happened in 2016 in the Cayman Islands that set size limits for how big the fish you can catch, and what we call bag limit, how many you can catch, and a closed season, that those are what can be done to help increase grouper populations, And ultimately, if you do it well, you can have plenty of fish in the ocean for scuba divers to see and people just to feel good about, and still have a sustainable fishery. But you just can't catch too many. And that's what had been going on for so many years. And that's not just a story of Little Cayman. That's a story of the whole Caribbean. That's really not a good story in my mind, that there's been so much decline of Nassau grouper throughout the Caribbean. But I think Caymanians on, you all on Cayman Brac and on Grand Cayman and here on Little Cayman should be justifiably proud of what's happened here, what the fishermen have been, participating in, and the conservation groups and the scientists from universities like myself and the Department of Environment, is that the Cayman Islands are, to me now, this sort of beacon of hope, if you will, for the rest of the Caribbean. That if the rest of the Caribbean can do what the Caymanians were able to do, we'll start to see Nassau grouper recovering in a lot of different places.
I also think that you guys are doing a big part of the work right now. Education is key. It's critical for protecting what we have out here. And so just knowing about the Nassau and you guys being a part of this and being able to share that information with your friends and your families is also a huge part of how we protect what we have. And this is, one of your greatest natural resources. And one of the challenges with a resource being underwater is that you don't see it all the time. And so us being able to bring it to you in your classrooms, for you to see it, it makes it much easier to protect that you've seen. Yeah.
And I would say the education shouldn't stop with you. Share the story with your friends and family.
Yeah. Yeah.
I think that's really important. I see there's another couple questions.
Yeah. So let's see. We've got Cayman Prep has a question. What are the maximum and minimum depths of aggregation sites? Great question.
Great question. And it varies from island to island. So where we're working here at the west end of Little Cayman, I would say that the maximum depth is about 130, 140 feet.
That's deep.
It's pretty deep.
Do you guys go that deep?
We don't.
Okay.
We only go down to about 100 feet. It's not safe for us to dive below that with the way we're diving right now.
Okay.
And then it maybe comes up in where the top of the fish are when they're in that big aggregation, maybe 60 feet deep.
Hmm.
If we were to go over to Grand Cayman, it sort of starts at about 150 feet. They're much deeper there, which is one of the reasons why it's very difficult to work over there. And Cayman Brac is in between, where it sort of starts at about 195 feet and maybe goes down to 130, 140.
So this is my guess. I'm going to hop into with a question here, you guys. Do Nassau typically live outside of when they're spawning? Do they live at those depths typically, or
It doesn't seem to, I mean, some of them do for sure,
But
but
they're
usually
typically
it's sort of 100 feet and shallower.
So why are they at that depth?
Because they like to spawn right on the edge of the shelf.
I see.
Right, and so and it turns out that right at the edge of the shelf on Cayman Brac is 130 to 150 feet.
Mm-hmm.
Here it's 100 to 120, and on Grand it's 150 to 250. So it just depends on what we call the underlying geology of the site, how it's shaped.
Right.
Yep.
Okay. Excellent. It looks like we have another question from CIS, and that is, how much has the size of the grouper population in Little grown since you started the project?
It has almost quadrupled in size.
Quadrupled.
4 times bigger than when we started. When we started, it was really not doing well.
Yeah.
It was maybe, we think, 1,800 fish.
Yeah, not
2,000 fish maybe, and now we're approaching 7,000. We think there's at least 7,000 fish out there this year.
Wow.
So it has really grown, and for an island of Little Cayman size, 7,000 big Nassau grouper.
That's big.
That's a lot, right?
Yeah.
And as best we can tell, it's going to continue to increase at least for a while.
Awesome. It looks like we have another question here from Cayman Prep, and that is, how do they minimize the losses from predators? And I'm assuming you're meaning, how do the Nassau minimize? I'm not sure.
Well, so what I think the question there is asking is how do they avoid predators? Certainly the government can't do that.
Right.
Not without really manipulating the ecosystem, right?
Sure.
They'd have to kill the predators, and I would say that's a really bad idea.
Right, because we know that predators have a really important
They play
top-down
such an
impact.
important role in the system, right?
Right. You actually want to see more predators out
Right,
there.
and we actually are. That's one of the cool things about this. I don't know if you had a chance to share that or not.
No, tell us.
But I feel like we are seeing more reef sharks out there as the population grows.
Wow.
And that's okay. That means there's more fish to feed the sharks.
Yeah.
There's more fish to produce eggs.
Yeah.
And that's okay. And as you just said, they're a really important part of the ecosystem. They're part of the predators are an important part of the fish community. And so under sort of what we consider natural conditions, they eat some Nassau grouper.
Mm-hmm.
And from a population perspective, that's okay.
Right.
And we do see some fish out there sometimes that have survived shark attacks.
There is a fish known as Lucky.
Lucky.
Yeah.
Yes.
And I'll actually post a picture of Lucky for you guys on the blog, and you can see. How do you know that Lucky was so lucky?
Well, because normally you have a fish and they're kind of shaped fish-like like that. Let me show my hands on the screen here. They're sort of fish-shaped like this.
Right.
And if you look at Lucky's back, it does that. It's got a big chunk taken out of the back of it. But he survived, which lets you know that these fish are pretty robust. They're pretty
tough.
strong. They're pretty tough. And
Yeah.
so if you can survive having a big old chunk taken out of you by a shark
And still show up
and still show up to spawn, they're pretty tough fish.
Wow.
But some of them do get eaten, and again, that's okay because that's just that's how nature works.
Right. We have another question from John Gray, and the question is, can an aquarium be built to keep groupers safe so that they can reproduce?
That's an excellent question. And in the fisheries world, we actually refer to that as something called stock enhancement. The stock is the population of fish. The enhancement is trying to help them increase in numbers.
Right.
And the answer to that question is yes and no. It can be done.
Mm-hmm. Has it been done?
And it has been done on a small scale, at least.
Okay.
Some researchers in Florida in the 1990s did it. I think there's some folks at University of Miami that are trying to do it now. Okay. And it's true that they can keep the adults safe and they can spawn them, although it's hard to spawn very many. As Dr. Simmons was talking about, we're talking about tens of millions of eggs being produced out here naturally.
Would be hard to reproduce.
I can't imagine building an aquarium that big, right? But you can. But the problem is that then maybe you grow up the babies till they're this big or something like that.
Sure.
That part of the work has been done as well. And so they release the babies into a nice seagrass area or something like that. They all get eaten.
So why do they get eaten?
Because they don't know how to behave in nature, right? Like if I had a guinea pig that I raised or something like that, and I let it go in nature, I think his odds of survival would not be very high.
Right, like my cat would not be able to go out in the woods and fend for itself very well.
So, but people are working on it, right? And so the answer is not no. The answer is it's really hard.
Right.
And so to me, the best way to make sure that we have grouper in the future is to make sure we have wild populations, wild healthy populations of grouper right now.
Awesome. So guys, we have time for some questions, and I would love to unmute some of you and have you come up to the camera and ask a question. So if you have one, you have Dr. Happel's full attention right now, and you can go ahead and unmute and ask a question. Or if you're feeling shy, you can always have your teacher type a question into the chat on the side. So while we're waiting for that,
do you want me to get the sampling bottle?
Let's do that.
Talk about that?
Yeah,
I'll be right back.
Okay. All right. So it looks like, David, were you gonna, Mr. Connolly, did you have a question that you wanted to ask or someone from your classroom? Okay. Scott is going to share one more pretty awesome scientific tool that they're using out here with you guys.
That's right.
Yeah.
So I assume that most of you at some point in time have heard the word DNA, right? It is our blueprints for our bodies.
Right.
And it tells us, or tells our cells what to do.
Right.
Right. And it's got all that information in it. Well, we use DNA and do DNA research for a lot of different things. And one of the things that we do DNA research for is to tell how populations are related, right? So we have this question, for example, are the fish on Little Cayman related to the fish on Cayman Brac?
Thanks for being here, John Gray. It was awesome to have you guys on. We're going to keep going for a little bit longer, so if you're able to, you can stay on and keep listening, but if you have to go, I completely understand. Thanks so much for being here. All right, so DNA.
Okay, so DNA. And so as it turns out, normally, to collect DNA, you have to have the animal, right? You might take maybe just a little clip of its fin.
Sure.
For humans, they might pull out a piece of your hair, things like that. And then they can tell, like if I had a piece of hair lying here, I could, in a DNA analysis lab, I could tell whether or not that piece of hair belonged to Todd Bohannon or not, right?
Right.
Because, and it's something that's fallen off of him. I don't have to sample Todd.
Right. You don't have to take a piece of me.
Exactly.
Right.
And so there's been a lot of advancement in science in the last 10 years on something what we call environmental DNA or eDNA.
Environmental DNA.
Correct.
Okay.
And so what it means is just measuring DNA in the environment. Right? So we have these thousands of fish out there, right?
Sure.
And they're swimming around, but they're doing other things, right? And they've got this mucus coating on their bodies, and things like that. Well, it turns out that all animals, what we call slough skin cells, they fall off, right?
And we have skin cells that are falling
slough off all the time, right? Especially
skin.
if you get a sunburn.
Sure.
Right? Yeah. Okay. And so these fish slough off their skin cells. And so we're working on a new project for what's called environmental DNA, where instead of having to handle the fish, we're going out and we're collecting water samples, right?
Yeah.
And we bring those samples back into our bedroom laboratory, as you talked about.
Yeah.
And we filter them through on a little tiny piece of filter paper. And then we're going to go back. I'm going to go back to my university and give those pieces of filter paper to our DNA expert. And she is going to then analyze those for grouper DNA. Just collected out of the water.
Just you're going out to the aggregation site, collecting water from maybe at the depth that they're at?
Yeah, around 70 feet deep.
And then you can take that and it's within that water that you've now captured, there's
grouper DNA.
Cells from the grouper that have sloughed off that are just in the water itself.
Right. Yeah.
And you can study that.
Yeah, we can. And these techniques are being used in not just in the ocean, but in rivers and streams and lakes all around the world. And they're being used to discover new species.
Wow.
Because you imagine we wanted to go work in the Amazon, right?
Sure.
It's a huge river, but it's super muddy.
Yeah.
You and I couldn't see each other if we were in the water right now. But I could take a water sample and bring it up and look at all the DNA from all the different animals in the water and tell what's there.
So literally, pretty much everything that's living in there leaves some sort of DNA fingerprint.
Right. And with modern DNA technology, we can detect that.
Wow.
Right? So I did want to show this just because it's kind of fun.
Yeah, here, I'm going to back up so we can see what you got.
So this is kind of a cool thing. We call it a Niskin bottle. And it is a water column sampler. It's a big tube, and it has two end caps. What I'm going to do, I can set this here, is show you how it actually rigs and works. So it's this big, long tube. And it has two end caps on it that are spring-loaded. So I'm going to pull one out there, and it's got a nylon line, and another one here. And I always trigger it at least once or twice accidentally while I'm doing this. But we basically clip these two lines together and put it in a little latch right there.
I see.
So now I have a rope and a line, and I can lower this down to whatever depth I want. And then when it gets to the depth I want, I clip a weight onto the line. Like I'm in a boat, right? I clip a weight onto the line and I let it go. And it comes down and it hits this little white button on top. Okay, so I'm going to be the weight.
Okay.
Here we go. Ready? So I've just dropped this down. Of course, the water is flowing through the tube all the way down.
Sure.
So it gets to depth. Drop the weight, hits this button, and water sample captured.
Wow.
So this is a really popular oceanographic device that has been used for a really long time to collect water samples to measure things like dissolved oxygen or salinity, things like that, boring stuff. Now we're using it to measure DNA.
Awesome.
And we'll be able to actually track changes in the population based on the DNA from a water sample.
That's really neat.
Super cool.
Yeah, I mean, that seems fairly revolutionary, really.
It really is. And as far as I know, we're the first to be able to try to do this for something like a Nassau grouper swallowing aggregation.
Wow. Yeah. Well, I can't wait to see what happens with all that
Me
data.
too.
We have one more question here from Miss Amanda and Miss Sherilyn's classroom at CIS. How long does it take for a grouper to go from its regular colors to its spawning colors?
Oh, also an excellent question.
Yeah.
And the answer is really fast.
Yeah.
It's amazing. So most people are familiar with the Nassau grouper with those really striking, beautiful barred patterns. Right. But we see actually 4 color patterns in Nassau grouper. We see that barred pattern. We see they can go almost completely dark.
Right.
They can go almost completely white. And then they go to what we sort of bicolor, which is half and half. They're dark on the top and they're white on the bottom. And they can go between those different colors. They could go through all 4 of those different color phases in 30 seconds.
So almost like an octopus.
Almost like an octopus.
Wow.
They can just change colors super fast. And it's what we call ephemeral, right? That means it just for an instant and they can change.
Yeah.
Right? But then they can change back.
And wow, that fast.
Yeah.
That's so great. Let's see here. If anyone else has questions, Cayman Prep High School, Shed, or Spot Bay, now would be the time as we are just about wrapping up. Do you guys there at the high school have a question up front?
Yes. Last night, a grouper had a pufferfish in its mouth, and he wanted to know what happened to it.
Yeah. So there, how did you know about the puffer that ate the grouper that ate the puffer? I haven't even shared that video yet.
We
Did you
saw
hear
it
about
on Facebook.
Well, I'm going to post a video about it, but maybe you
Yeah.
could tell what happened.
I don't know. I would love to know. We haven't found out yet. That's
But what will happen?
What will happen? So, fortunately for the grouper,
Yeah.
if it has something lodged in its throat, unlike us, it can still breathe,
Right.
Because it can breathe through its gills.
Right.
So as long as it can continue to kind of crush down with its jaws onto that puffer fish, and eventually squish it flat, he'll be able to swallow it and eat it, but he'll be able to keep breathing, fortunately. So he won't choke.
Okay.
But how long that takes, I don't know. Maybe we'll get to see him again this week.
Yeah.
But I haven't seen him since.
We'll keep our eyes peeled for him, and I'll share on the blog for you guys the video of that grouper with the puffer in his mouth. It's pretty striking. I've never seen that before.
Yeah. That was the first time for me too.
Right?
Yeah.
Yeah, that's crazy. Any other questions before we head off for the day? All right. Well, I want to thank you guys all for joining us for these live feeds this week. It's been awesome talking with you. Please keep following along on the blog. I'll keep posting stuff today, tomorrow, and Saturday while I'm here on the island with the researchers. And if you have any other questions, you can post those on the blog, and I will respond to those that same day. Let's see. Hi, Spot Bay. Hi, guys. It's so good that you guys are here. Did you have another question, or are you done?
Yeah.
What's your question?
Will a grouper fight with its own kind?
Say that again.
Will a grouper fight with its own kind?
The question is, will a grouper fight with its own kind, with another grouper?
That's a most excellent question.
Yeah.
And the answer is yes.
What does that look like?
Well, they tend not to really beat each other up, but they tend to do this sort of mouth-to-mouth thing where they're like, rawr, rawr, and they try to talk it out. And sometimes they'll spin around each other's and look at each other. And the Nassau grouper mostly don't do it when they're spawning.
So when they're spawning, they get along.
They get along pretty well.
Any other time,
they're when they're at home, this is my reef.
Right.
You're not allowed here.
Sure.
Move along. Yeah.
And they'll change color when they fight.
And they'll change color when they fight. Yeah.
Wow.
So we talk about how fast they change. Oddly enough, the tiger grouper, which are also spawning out there, fight when they're spawning.
And I saw that when I was out there the other day. I'd never seen that before. And they were a little more active in their fighting.
And you'll see the big male tiger grouper, and they'll have big scrapes and cuts and things like that from fighting with the other males.
Wow.
Yeah.
That's incredible.
Good question.
Great question, Spot Bay. Do we have any other do we have a question from Shed Aquarium? All right. Well, nope. Okay. Thank you guys so much again for being here, and be sure to keep an eye on the blog. And thank you again so much. All right. Have a great rest of your day, you guys.
Thanks, everybody.
Take
nice
care.
talking to you.
All right. Bye-bye.